Thank you so much for inviting me and letting me participate remotely. I'm really excited to be here and have a discussion with you all today. Um, okay, does that look good? Great. Okay. So today I'd like to talk about kind of philosophically, the approaches that we take and what are some of the things that we might want to start thinking about when we're thinking about, how to manage tree populations under climate change. So a lot of evolutionary approaches in trees take what I like to call the methods triad, looking at genotype phenotype associations, genotype environment associations, and phenotype environment associations. And these types of analyses have given tree biologists insight into which traits are involved in adaptation to environmental stressors such as freezing, aridity, and disease, and have led to the discovery of some of the genes that are involved in adaptation. But the ability to translate this kind of genomic data into a specific prediction is still limited. And by prediction, I mean a prediction about how that population will perform under climate change, how vulnerable it is, or a prediction of how to better match individual genotypes to multivariate environments for seed transfer, or something like that. So today in my seminar, I'm going to highlight two areas of research that are important in determining how organisms respond to environmental change, but aren't commonly considered in tree biology. In part one, I will highlight how genetic bioenvironment interactions are not the only way that genotype and environment can be non-randomly associated. We can also have covariance evolve between genotypic and environmental effects on phenotypes. In part two, I will highlight how phenotypic clines can evolve without clines in the underlying alleles, and this has implications for our ability to understand the genetic basis of adaptation to environments. At this point, we don't really have a solution to this problem. I'm just going to talk about the problem and talk about what the next steps might be here. So the first part of this seminar will highlight our research into the evolution of covariance between genotypic and environmental effects on phenotypes. And this is in collaboration with Molly Elbecker, my former postdoc, and Jeff Trussell. And this research is funded by the Research Coordinated Network for Evolution in Changing Seas. I am also a marine biologist, but there are a lot of parallels between marine species and tree species. They both have long generation times, potential for high gene flow, large effective population sizes, and they're both threatened by really rapid climate change. So biologists have a rich history of studying clines, and clines often serve as an observational basis for studying adaptation. So for example, biologists go out, they might see that in one environment, there's a different phenotype than another environment, and that makes something interesting to study. And to try to understand what's causing this pattern, we do reciprocal transplant experiments or common garden experiments, and these experiments help us disentangle whether this clinal pattern is due to environment only, genotype only, or some kind of genotype by environment interaction. And we know that these genotype by environment interactions are common, and its genotype environment interaction is essentially a non-random association between genotypic and environmental effects on genotypes. And it ultimately means we need to have information about both genotypes and their environments to make any kind of prediction. But G by E interactions are not the only way that genotypic and environmental effects on phenotypes can be associated. We can also have covariance evolve between these genotypic and environmental effects on phenotypes. And I'm just going to call this COVE-GE for short in this talk. So this COVE-GE was first recognized by Levins in 1968 in Drosophila melanogaster. It was recognized again by Bervin in 1973 in frogs. and made popular by Conover and President in 1990 when they saw these patterns in silversides, which are these fish that adapt to freshwater, like estuary environments in the East Coast of the US. Now, the interesting thing about all of these classic studies is they didn't actually quantify this COVE-GE. They identified it by looking at visual patterns and reaction norms. So I'm going to show you the visual patterns look kind of like this, but the identity of the genotypes and the pattern across environments matters. So I'm going to draw your attention to this graph on the left here. This is an example of positive COVE-GE. So in this case, if we look at the average genotypic effect of genotype 1 from environment 1 and genotype 2 from environment 2 on the phenotype, we see that there's an increase. So the genetic effect is an increase. As we move from the environmental effect on the phenotype from environment 1 to environment 2, we see also an increase in the phenotype. So this is positive covariance between genetic and environmental effects on the phenotype. In contrast, we can also have, if we have a decrease in the genotypic effect from environment 1 to the genotype that evolved in environment 2, that's a negative G effect. But this environmental effect, the plasticity here is still positive. So here in this case, there's a negative COVE-GE because those genetic and environmental effects oppose each other. So on the left here, this is called cogradient variation. And if we go out and we survey these populations in their home environments under this cogradient variation, we would see a very steep climb. Under countergradient variation or negative COVE-GE, if we went out and surveyed these populations in their home environments, there might not be an observable phenotypic climb, but nevertheless, there's important genetic differences that underlie those populations. So there hasn't historically been a quantitative method to measure this. And it's only been based on this visual patterns that researchers have talked about this. And that visual detection has limits. It's not a statistical test. And that means that we can't answer bigger questions about covariance, such as, how common is it? Is it as common as G by E? And why haven't we measured it? So in 1989, Falconer pointed out in the Intro to Quantitative Genetics textbook that phenotypic variance is broken up into environmental variance, genetic variance, genetic by environment interactions, and this COVE-GE. And to our knowledge, no one has actually estimated this variance component. Falconer didn't really describe how to estimate it in the presence of residual variation and G by E, and didn't describe what to do if COVE-GE was negative. So to our knowledge, no one has actually estimated this variance component. So we were thinking, well, if we can't estimate the variance component, maybe we can estimate the effect size of the COVE-GE. And so we developed a sampling estimate to measure COVE-GE. And to walk you through the intuition for this, I've got our reciprocal transplant data here on the left, and I'm just going to plot the environmental effects on the phenotype versus the genotypic effects on the phenotype here on the right. So first we get the genotypic effects from the data, and I'm just going to plot them here on the right. Next, we get the environmental effects from the data. Those are plotted here now. And once we get those from the data, we can then measure the covariance between these genotypic effects and environmental effects. And this is the equation that we use to calculate the covariance between these effects. This equation gives us a number between -1 and +1. Negative is countergradient variation, or negative COVE-GE. Positive is cogradient variation. And you may, if you're familiar with statistics, you may recognize this as looking kind of similar to a correlation, but there's some important differences. So this is measuring the genetic effects. This is measuring the environmental effects. And you'll notice that we have this I variable. This is an indicator variable. And this just indicates that we only want to count effects towards the covariance when those genotypic effects are matched with their environmental effects. And then finally, you'll notice that we have a standardization that is a little bit different from a normal correlation. And we can't use a normal correlation here because the genetic and environmental effects are not independent of each other. And so to account for this non-independence, we standardize by one or the variance in one or the other, depending which is larger. And then finally, these genetic and environmental effects are not known with certainty. And so we use bootstrapping for 95% confidence intervals on COVE-GE and permutation to calculate a p-value against the null hypothesis that COVE-GE equals 0. So to validate this metric, we created 30,000 plus simulated patterns. We simulated reciprocal transplant experiments. Here's an example of a data set with 4 populations, 4 genotypes in a 4x4 reciprocal transplant experiment. The method gives a COVE-GE estimate, confidence intervals on that estimate, and a p-value. In addition, we added genetic bioenvironment interactions into our simulations, and data sets can have both G by E and COVE-GE in them. This is an example of one of those data sets. And going back to that history of this pattern being visual, this kind of pattern might be present in a data, but if people are focused on visual cues, they may not recognize COVE-GE, even though this is very strong COVE-GE. We also simulated paired common garden experiments. So for example, maybe you have trees adapting to altitude and you collect one pair of ecotypes from one part of the range and another pair of ecotypes from a different part of the range, but you only raise them into environments like a low altitude and a high altitude environment. We also can calculate COVE-GE from this kind of experimental design. So we conducted a power analysis to understand what kind of experimental designs would give us power to detect COVE-GE. So for reciprocal transplant designs, the number of genotypes will be shown here on the y-axis, and the sample size, the number of individuals from each genotype, will be shown on the x-axis. So 8 would be, an 8 population by 8 location reciprocal transplant design. The total sample size is shown on the top of each cell, and then the power of that design is shown on the bottom of each cell. So the first thing to notice is that you need really large sample sizes to have high power to detect COVE-GE. And the same is also true for the paired common garden designs. The other thing to notice is that reciprocal transplant designs, which are just a 2x2 design, have really low power to detect COVE-GE. We also applied this to empirical data. So here's an example from Molly, my postdoc's frog system. She had studied these freshwater tree frogs that were adapting to saltwater environments. And her pattern, the pattern in her data does show significant negative counter gradient variation. And we also applied it to some of Jeff's data, my other collaborator who works on snails, and had found these patterns of counter gradient variation in these low flow versus high flow environments. But it wasn't significant by our method. And that goes back to the fact that these 2x2 designs have really low power. So now we're in a position to ask, well, how common is this in nature? Is it as common as genetic bioenvironment interactions? So we done a pretty comprehensive meta-analysis. We started with 4,000 studies looking for search terms related to reciprocal transplant experiments and local adaptation. And at the end of the day, a few hundred traits of across about 70 studies met our criteria. So I'm going to plot the pattern, the initial pattern that we see in this meta-analysis on this graph. On the bottom x-axis here is the COVE-GE metric that I've been talking about. On the y-axis is a standardized metric for genetic bioenvironment interactions. And here's the pattern that we found in the data. So as you can see, it's all over the place. we find evidence for COVE-GE in a lot of these systems, and quite a lot of data sets from trees met our criteria, which is great. But the interesting thing about looking into these studies is that a lot of people did not necessarily recognize that they had patterns of COVE-GE in their data, or it wasn't discussed as part of the study. So in terms of climate change and plasticity, what are we missing? This counter gradient and co-gradient variation could be an important source of non-random effects between genetic and environmental effects on phenotypes, and they may affect also how a population responds to climate change. So to further understand this, we're developing these simulations that are evolving this co-gradient variation and this counter gradient variation. And so the first step is to understand how does it evolve? And we're still working on that. And then after we understand that, we'll be able to change the climate in the simulations and understand how does a co-gradient versus counter gradient pattern affect how a population responds to environmental change. So to conclude, adaptation and climate change, what are we missing? In the first part of my talk, I highlighted how covariance can evolve between genotypes and environments in a non-random way. And if we're focused just on looking for clines in natural populations, we may be missing important traits that have this negative co-GE because they would be less likely to show strong clinal patterns on the landscape. And then secondly, I highlighted how considering adaptation in more complex environments could evolve phenotypic clines without clines in the underlying allele frequencies. And my lab is working on solutions to this. I think some solutions may be in machine learning or other kinds of multivariate analyses. So if you're interested in this, I am looking for PhD students to work on these problems. So with that, I would like to thank the National Science Foundation, who's been funding this research. Thank you for listening, and thank the organizers again for inviting me. And I'm excited to have a discussion with you all about this research.